Hello, I'm Amir, and this is my YouTube channel, Genetics with Amir. The topic of this video is about CRISPR-Cas9, which is considered a revolution in genetic engineering. CRISPR-Cas9 is an acronym for Clustered Regularly Interspaced Short Palindromic Repeats. So, what's the meaning of this? I'll explain it to you on next slides, and at the end, I'll review this name with you. This genetic engineering technique is modeled after a process found in 45% of bacteria and 87% of archaea. While studying a gene in E. coli at Osaka University in Japan, researchers first observed repeated and non-repeated sequences in proximity to this gene, but their biological significance remained unclear for 20 years. These sequences were eventually recognized as a component of the bacterial adaptive immune system. A sequence found near the repeated sequences, identified as Cas, was found to be crucial. Its absence resulted in the inactivation of bacterial adaptive immune system. As you know, the immune system is divided into two branches, the innate immune system and the adaptive immune system. As previously mentioned, CRISPR-Cas9 is part of the adaptive immune system. Cas stands for CRISPR-associated protein. The Cas protein is actually an enzyme. For example, Cas1 and Cas2 together form a hexameric enzyme complex. This enzyme complex has two main functions. 1. Cutting a specific part of the invading DNA that enters the bacterium for the first time. 2. Transferring the cut DNA to the CRISPR sequence. But exactly where on the invading DNA does this cut occur, and where in the bacterial CRISPR sequence is it inserted? Let's break it down. As I mentioned in the previous slide, the hexameric enzyme complex Cas1 and Cas2 cuts the invading DNA that enters the bacteria and inserts it into a sequence region called the CRISPR array. CRISPR array. This is a region where the spacers and repeat sequences are located. Each spacer is the DNA of a specific pathogen that has inserted its genome into the bacteria. The bacteria, using the Cas1 and Cas2 enzymes, insert this DNA into its own DNA. Whenever the bacteria encounters that specific pathogen again, this sequence is transcribed and the bacteria becomes ready to fight it. Repeat. This is a palindromic repeated sequence that is located between the spacers. All repeats have exactly the same sequence. Now let's look at the figure to fully understand what I've said. For example, this red pathogen inserts its DNA into the bacteria for the first time. The hexameric enzyme complex comes into action and cuts a specific part of the pathogen's genome and inserts it into the bacteria's genome in the CRISPR array region. It's important to note that the new spacer is always placed at the beginning of the CRISPR array region. By doing this, the bacteria is ready for the next encounter with the pathogen. Now let's assume the red pathogen attacks the bacteria for the second time. This time the bacteria transcribes from the CRISPR array region and creates pre-cRNA, which is an immature cRNA that undergoes processing and becomes cRNA. This RNA enters an enzyme called Cas9 and goes and cuts the pathogen's DNA, protecting the bacteria. I'm going to add more details to my explanations each time, so you can learn more deeply and accurately. Okay, in this slide we have another opportunity for review. A pathogen attacks the bacteria and inserts its genome. The bacteria integrates a specific part of the virus genome into its own genome. Then, if the same pathogen attacks the bacteria again, the bacteria begins transcription from the CRISPR array region, producing pre-cRNA and ultimately cRNA. Additionally, an RNA called tracer RNA, which is transcribed from another part of the bacterial genome, comes into play, as does the Cas9 enzyme. Finally, these three components, cRNA plus tracer RNA plus Cas9, together form a complex called the effector complex, which binds to a specific sequence of the pathogen, cuts it, and destroys it. In this figure, we see the promoter and the protospacer. The protospacer is described as a specific region that the Cas1 and Cas2 enzyme complex cuts in the pathogen and inserts into the beginning of the CRISPR array in the bacteria. This is a key point. It clarifies that the protospacer and spacer have similar sequences, but the protospacer is located in the pathogen, while the spacer is integrated into the bacterial CRISPR array. So, I have a question. If the protospacer and spacer sequences are similar, why do Cas1 and Cas2 and Cas9 only cut the protospacer? This is a central mystery in the slide and I want to explain it. It is because of trinucleotide sequence called PAM. which is short for protospacer adjacent motif. This sequence exists upstream of the protospacer sequence, but does not exist in the spacer, and this prevents the enzyme from cutting the bacteria's spacer sequence. What we mean by NGG is that in the 3 nucleotide PAM sequence, the first nucleotide can be any organic base, but the second and third bases are guanine. This slide illustrates how the Cas9 enzyme binds to the target sequence at the protospacer region. Cas9 cleaves the DNA before the 3 nucleotide PAM sequence. Cas9 has two crucial requirements for cleavage. 1. Base pairing between the cRNA sequence and the protospacer sequence. This refers to hybridization between the cRNA and the protospacer sequence in the target DNA. This ensures that Cas9 targets the correct location. 2. Presence of PAM upstream of protospacer. In this slide, we see that RNA polymerase transcribes the CRISPR array, and then tracer RNA binds to the part of the repeat sequence. Then, the RNase 3 enzyme causes processing in the pre-cRNA. Finally, the cRNA and tracer RNA remain attached to each other and enter the Cas9 enzyme. Note that the role of tracer RNA is a hook that keeps the cRNA within the Cas9 enzyme. cRNA is also called guide RNA because the enzyme uses this sequence to find the target sequence in the pathogen. Dr. Jennifer Doudna and Dr. Emmanuel Charpentier in 2012 they invented this method, and finally in 2020 they won the Nobel Prize. An interesting point in Dr. Doudna's name is that the 3 letters DNA can be seen, and it's as if working on these matters is in her blood. What is the difference between the invented CRISPR method for genetic engineering and the CRISPR system that exists in bacteria? In the invented method, instead of cRNA and tracer RNA, a sgRNA, which stands for single guide RNA, which is a synthetic RNA molecule, is used to recognize or target sequence. What are the uses of CRISPR in genetic engineering? Oncology. 1. Removing or turning of oncogenes. 2. Activation of tumor suppressor genes. 3. In vivo deletion of non-G and non-G P8 genes, which are involved in prostate cancer, and the result was a reduction of tumorigenesis in mice. 4. At Sichuan University in China, they are trying to equip human T cells with specific receptors for the treatment of multiple myeloma, sarcoma, and melanoma by ex vivo genetic engineering. Genetic diseases, editing in HBV gene, viral disease, delete HIV in cells, and cure bacterial infection. Innovations in CRISPR. It is not only used for gene cutting, but the enzyme can be combined with inhibitor or activator and affect the expression of genes. Base editing. Chemically and without breaking the DNA, we can change the type of base. Prime editing. Insertion and deletion without cutting the DNA strand. Finally, know that we have given an overview of CRISPR-Cas9. As I mentioned in the first slides, I revisit the meaning of its long name. CRISPR, acronym for clustered regularly interspaced short palindromic repeats. This means that short repetitive sequences are regularly spaced between spacers, and all of these parts are located close together. Thanks for watching.